Good afternoon, everyone. My name is Florian Plaza Oñate. I am a bioinformatician at Metagenopolis. And today I would like to talk to you about CroCoDeEL, a tool we have been developed to detect cross-sample contamination in metagenomic data. So I assume that most of you are familiar with metagenomic sequencing. It's a very powerful technique to study a microbial ecosystem, but it's subject to many technical and experimental biases, including contamination, which is defined as an unintended presence in a sample of DNA from sources other than the biological material under investigation. So there are two main kinds of contamination. So the first one is external contamination, when you have in your sample, for instance, DNA from lab reagent or laboratory equipment. But today, this is not the kind of contamination I would like to talk to you. I would like to talk about cross-sample contamination, which is sometimes named as well-to-well contamination. And is the intentional transfer of DNA from one sample to another. In other words, it's accidental mixtures of two samples processed simultaneously. So it can occur during DNA extraction or sequencing library preparation, and it has several identified causes, such as pipetting error or formation of aerosol and microdroplets while removing films for sealing plates, for example. So this kind of contamination is problematic because in your contaminated samples, you will detect microorganisms that are actually not present in your sample. So they are forced detection of microorganisms that can be, for instance, pathogens. It will artificially increase alpha diversity indices, in particularly microbial richness. And when you perform a strain sharing analysis, you can detect samples that share strain, but that actually do not. So it compromises all your analysis and eventually the conclusion you get. And it's particularly critical in a clinical context when you analyze each sample individually. So we need methods to detect this kind of contamination. And the most known method is negative controls, which are tubes or wells with sterile purified waters. And typically, when you detect DNA in negative control, for instance, using PCR or sequencing, it reveals that contamination occurs. So negative control is essential, a very powerful technique to detect and eliminating external contamination, but it's limited to detect cross-sample contamination. It can reveal that cross-sample contamination occurred during lab processing, but it cannot detect cross-sample contamination between real, meaning non-controlled samples, because the contamination sink, is necessarily the negative control and not a real sample. Another way to detect cross-sample contamination is to perform strain-level bioinformatics. So you perform a strain analysis in all your samples, and if you detect unrelated samples that share strains, it may indicate cross-sample contamination. This technique is very powerful and has been proposed by the Banfield labs in the paper I just show here, but it has several limitations, and one is that it does not work for samples that may naturally share strains, such as, for instance, the fecal metagenome from a mother and a child, because you don't know if it's contamination or natural vertical transfer. And it has other limitations. So this technique does not directly distinguish when you find two samples that share strains, which one is contaminated and which one is the contamination source. And the estimation of contamination rates are not straightforward. And in addition, strain level analysis can be a computer intensive task. And from the best of our knowledge, there is no ready-to-use and easy user-friendly tool available to apply this method to other datasets. So this is why we developed CroCoDeEL, a new method to accurately detect cross-sample contamination in shotgun metagenomic data. So it can detect cross-contaminated samples, including non-controlled samples. It does not need negative controls. and identifies clearly for all contaminated samples their respective contamination sources and provides an estimation of contamination rates. So as I say, it does not need the negative controls, and you do not need to provide external metadata such as the sample position on the plates if you work with plates instead of individual tubes. And it's based only on species abundance tables, so you can use easily with your pipeline and you don't need to do a lot of changes. And finally, it's way more sensitive than strain level approaches. So here I will show you how CroCoDeEL works, so just pay attention. So here we consider a pair of two unrelated samples. So we generate a taxonomic profile for these two samples. So each sample is a column, each row is a species. Here you have the relative abundance of all species. And you can visualize this in a scatter plot here in log scale. So we compare the abundance of all the species in sample one here and sample two here. And sorry, what's going on? Okay, so as you can see, when we compare the species abundance profile of these two samples, there are species specific to sample one, species specific to sample two, species that are shared between the two samples, but there is no specific pattern. So what happens when cross-contamination occurs? We can simulate that by mixing reads from these two samples here in the 90 and 10 proportions. So we have the two samples, the sample before contamination, the contamination source. We take 90% from this one, 10% from this one, and boom, we get simulated contaminated samples by mixing reads. Then we can generate the taxonomic profiles again. So here is what's before contamination, what I showed you before, and here is what we can see after contamination. So as you can see, there are specific patterns that emerge. So first, we can see that above a given abundance, all the species from the contamination source are seen in the contaminated samples because there are no more species on the y-axis here. And there is a pattern here we call a contamination line, which is formed by species that were not present in the contaminated samples before contamination. And these species have a proportional abundance between the contamination source and the contamination sinks. So this particular pattern, the contamination line, is the thing we use to detect cross-samples contamination. And we can, by focusing on the abundance of these species, estimate the contamination rate. Okay, so the human expert is very efficient at detecting such contamination pattern, but such task is actually error-prone and time-consuming because, for instance, for 96 well plates, you'll have to check for 5,000 profiles, so it's very time-consuming. And among all the profiles you have to check, you will have only a few positive cases. This is why we developed CroCoDeEL to automate this task. So CroCoDeEL can be seen as a black box. It takes as input species abundance profiles, and then output you a TSV file listing all the suspected contaminated samples, their respective sources, the estimated contamination rate, and a confidence score. So how CroCoDeEL works, I have no time to give a lot of details, but basically it takes all the sample pairs in your species abundance table, extract features that have been deemed relevant by human experts. So for instance, we use linear modeling to search for a contamination line. We count how many species are on this potentially contamination line. And then we provide all these features to a pre-trained random forest model that will indicate if the pair of interest is potentially contaminated or not. So to check that CroCoDeEL works works and can detect cross-sample contamination, we perform several benchmarks. I will show you only one in this presentation. So we generated 8 intentionally contaminated samples by mixing purified DNA from real human fecal samples. And we have tested it with CroCoDeEL, and CroCoDeEL finds all the contaminated samples, including those with very low rates. So you have, for instance, here 3 cases. On the y-axis, you have the contamination sources. On the x-axis, you have the contaminated samples. On all these cases, you can detect you can see the contamination line, including this one where the expected contamination rate is very low. So it provides accurate estimation of contamination rates. As we can see, for instance, in this example, the expected contamination rate is 10% and the observed is around 11. And we have not detected false positive. So the high sensitivity and specificity are confirmed on the large simulated and real data sets, and they are shown in our preprint if you are interested. So as a conclusion, this is something I have not shown in this presentation, but it's shown in our paper. Cross-sample contamination is actually a widespread issue in metagenomics, but remains overlooked compared to external contamination. And we do believe that cross-sample contamination should be a mandatory quality control steps in metagenomics. And so we have proposed a tool to detect such contamination. It does not require negative controls, and it's actually fast, so you can process 100 samples in a few minutes on a regular server, and it works with limited computing resources, including RAM. And you can apply it to any data sets, including those that you found on the public repositories, such as NCBI, ISRA, or the European Nucleotide Archive, and you don't need external metadata, such as samples positions or sequencing data coming from negative controls. CroCoDeEL is open source software. It's available on GitHub. You can easily install it using Conda. There are also containers available for Docker and Singularity. And if you're interested, there is also a preprint available in bioRxiv where we provide extensive benchmark with semi-simulated and real data sets. And we provide applications cases on published studies, and we show that some conclusion of these studies may be false or partially at least. So I would like to say thank you to all my colleagues that were involved in this project. And if you have any question, I would be happy to answer. Thank you for your attention.
Thank you, Florian. So in the interest of time, I'll take only one quick question. If someone has a quick question for Florian, there's, yeah, Christopher.
Through the microphone, please. Okay, so cross-sample contamination is an important problem and definitely happens. But when it does, it's most often barcode switching, in my experience. And that's going to mix up more than a pairwise samples. If barcode switching is happening on a run, your samples will be composed of contaminants from multiple other samples. And I don't see that your method can identify that.
Well, actually, if you work with individual tubes, cross-sample contamination can occur as little chance to occur during lab processing. But when you work with plates, it happens a lot because the physical separation between the wells is tight. CroCoDeEL can detect contaminated samples when there are multiple sources. From the best I know, it can work with 2 or maybe 3 different contamination sources, but when you have tens of them, it won't work properly because the method works if contamination introduces specific species in your contaminated samples. So if you have many contamination sources, the method won't work.
Yeah, personally, I think that is the most likely scenario. Anyway.
No, actually, when you work with plates, you can have a lot of cross-sample contamination because, for instance, you seal the plates with a film, you remove the film, and there are microdroplets that are spread everywhere.
Okay. All right, fair enough.
All right. Right, thank you. Sorry, I have to cut it off here. I'm actually going to give it over to